So we have now gone live. Welcome for those of you who are tuning in to the third Artificial Life seminar, virtual seminar, the third of the year and the third of all of them that have taken place so far, as this is the first year of them. It's my real pleasure to have Martin and Silvia here to be presenting some of their work. Martin and Silvia are both based at the University of Trento in northern Italy. I had the great pleasure to visit them there, gosh, about a year ago, just under a year ago, and it's a great pleasure to have them here and to have the opportunity to listen and talk about their work. I've started a little bit of a tradition with these seminars of asking our speakers to, before they dig into the academic content of their talk, to just briefly mention the first Artificial Life conference that they went to, and perhaps something that happened at it or somebody they met that was interesting. So if you don't mind, Martin, if you're up for kicking this off with a little blurb along those lines, that would be much appreciated. What was your first ALIFE conference?
Great. I think you were there too. It was at the University of Leicester in the UK, but it was not organized by the University of Leicester. It was by a different university. That's why I'm a bit confused which one that was.
That's right.
Do you remember?
Yeah.
I met you there for the first time.
Yes, that's right. And it was Seth Bullock that was organizing it, if I'm not mistaken. That's right.
Yeah.
So that was Southampton that was organizing, I think, but it wasn't in Southampton, just as you're saying. Yeah. Yeah.
So I was convinced by my collaborator Takashi Ikegami from Tokyo to present for the first time at an ALIFE conference, because he said that the things we were working on would be of interest to the community, and I didn't know anything about really the ALIFE community, although I knew several key people. I've never been to one of the conferences, so it was terrifying to go up in front of these immensely smart people and talk about my chemical system, and I didn't know the terminology to relate to them, so I had to really go in blind. And they were very supportive. It was a very supportive feeling that I got from the crowd, even though I had to learn to speak their language.
That's great. Yeah, and I think the rest is history, in the sense that I think Martin's work is pretty well known now in the ALIFE community as an example of wet ALIFE and connecting the ideas in the artificial life community to actual lab-based research. And so with no further ado, I'd love it if you could just take it away. And so Martin and Silvia are both going to be presenting. I think Martin's going to speak first, and then Silvia will join in partway through.
Yep.
Thank you. Thanks again for presenting, and I look forward to your talk.
Thank you. Thank you for inviting us. I will start sharing my screen, which has some slides here. Oops, we're in the wrong place already. So you should see my title slide. Is that correct?
That looks good from my end. Yeah, looks good.
Okay, great. So thank you for having us here. Silvia and I have been working together for quite a few years on chemical droplets and their relationship to artificial life community and artificial life sort of questions. And we're going to give you some, I will give you more of an older person's introduction to what's going on with these droplets and why they might be interesting. And then Silvia will give you an update on the most recent stuff that we've published, what we're actually doing with these. So basically, I'll be talking about the liquid droplets. Let me get my pointer out here, laser pointer. I'll be talking about liquid droplets. And Silvia then will take over and talk about how we're using them as transporters, interfacing them with living cells. Okay. So this is very old and probably no longer used definition of synthetic biology, but basically this is the one that kind of guides much of the work that we're doing, where we try to reproduce elementary vital phenomena of living systems. So starting simple and then building to be more complex through a bottom-up approach. And so one of the things that we found was useful was to use simply liquid and liquid systems. So make droplets. It's very easy to make droplets in your kitchen. It's just as easy to make droplets in the laboratory. So we thought that was a nice system to start to work with, especially because we're interested in one of the phenomena of self-motion, as you will see. So this is just an image of a droplet here, and how this might relate to artificial life. There are several ways that we can push these systems. We will be talking about, Silvia and I will be talking about movement and chemotaxis today, how you get these things to move and how we can control some of the motion of these things. If we push on that more, we might be able to say something about agency from a sort of chemical embodied perspective, maybe something about memory, how that could be embodied in the systems, and maybe something about the origins of cognition, which is really far away from just considering the chemistry of these droplets, but still we try to make some sort of path towards these more interesting phenomena of living systems. At the same time, we can push the dynamics of these in another way towards division and fusion, and we've done this already, maybe leading eventually to replication cycles. And if you can incorporate replication cycles with some sort of substantiation of information, maybe towards evolution eventually of these non-living chemical droplets, basically. So how far can we really push these towards somehow representing the vital phenomenon of living systems? That's where we're going here. So we'll stay here today, this part of the scheme. I will present to you now a video. We have a few videos that we prepared. They're all on the macro scale, so you can see them with your eye. You don't need a microscope, typically. And they're typically put in a glass dish, as represented below here, filled with some aqueous phase, and then we add an oil droplet to it. And there are two different systems Silvia and I will talk about. I will speak about this one, where basically the oil droplet, it has a body which is nitrobenzene. And for those of you not so interested in chemistry, all you need to know is this is non-reactive. It's just a droplet that you put into water, and if that's all you have, it will do nothing. It'll sit there and will not mix. In order to animate this system, we have to add some sort of fuel. So this is onboard fuel. So we mix in this chemical precursor, oleic anhydride, with nitrobenzene. These things mix very well, and they form these droplets that are then placed in the system. And the environment is this glass dish here, which is pretty big. It's a few centimeters in diameter, with some aqueous phase, some water and some soap, such as oleate here, is put in the system. So it's a system that is charged. It's charged with chemical potential. There's a reaction here. And this is what the output looks like. I'm going to play a video which might be choppy on your end, so you have to kind of imagine that it's a smooth motion, because it actually is very smooth motion. I put a red droplet in this dish, and it starts to spontaneously move around. It breaks symmetry and starts to just self-propel itself around like a little rocket, let's say, inside of this this blue dyed pool, and all of the chemical details are up in the right corner here. So if you want to see that, and this is nice because it spontaneously breaks symmetry, and it has its own motion, which we can study. And the other thing that's nice about it is we can see that there are chemical trails that you might be able to see coming up behind there. The chemical trails are acidic trails, so the chemistry is producing acids, and we could actually monitor that in real time using these kinds of different reference dyes and things. So we can get this thing to move. Very simple composition. Oops, I've lost the screen here. Oh, here we go. Very simple composition chemically, but so easy to make. Here's the chemical potential. So this is the fuel. It's an anhydride. In the presence of water at high pH, it's hydrolyzed to oleate, 2 oleate molecules down here, and protons. The protons are what's causing this pH gradient that you saw in that blue pool. So it's basically making acids as it goes, and surfactants. Okay, but that's the very simple metabolism. Doesn't require an enzyme or anything like this. It's just a downhill reaction that is just going on and on. Eventually, it'll burn up its fuel, and it'll stop in the system. And what we realize is if we represent the droplet here in the center, that these molecules will self-assemble at the interface between the oil in the center and the water in the environment. And these self-assembled molecules, this will get into more of the chemistry here. It's not so important that you understand the chemical details, but I'll just say it briefly so I could fairly represent the system. When these molecules assemble at this interface, they have two states. One is a protonated state on the right, or a deprotonated state on the right, or a protonated state on the left, depending on the pH. So if the pH is higher in this state on the right, which is deprotonated and charged with these blue amphiphiles sort of assembled at the interface. And if the pH is low, the surfactant is protonated, still at the interface, and that's just kind of represented by these red ones here. And this is a simple switch. It's a chemical switch, and it depends on the pH. And if you looked at the graph below, you'll see how the tension, interfacial tension at that droplet changes depending on which pH you're in. So we typically start an experiment at pH 11 here, and it produces acid, so the system is going down in pH. And when the system goes down in pH, the interfacial tension along that droplet starts to rise, okay? And normally that's not a problem. It's not a problem at all, and the situation would do nothing except the fact that this thing starts to move and break symmetry. It's because one part of the droplet is experiencing a different pH, like low pH here, versus this part of the droplet, which experiences high pH. Therefore, the tension along that interface is much different, and it's basically giving an impetus for the system to do something to equilibrate that tension. It doesn't want this difference in tension along this. It needs to go to equilibrium. So it starts to move things around to try to compensate for that. And when it moves things around, it basically forms a convective cell. This is the motor here. So this video, which should be a smooth running video, I don't know if it's smooth on your end, but believe me, it's a smooth running video where you see a droplet with some fluorescence. And when it comes into focus here, you can maybe see that through the center, there's a flow of material, and then along the interface, and then back through the center. This is typical physical system that has, in the presence of a gradient, then forms a convective cell. So this difference in tension along from one part of the droplet to the other part forms a convective cell because it's trying to equilibrate the tension differences. And by basically forming a convective cell, it keeps churning itself up, bringing fuel from the oil phase to the surface where it's hydrolyzed, forming a pH gradient, which it then tries to resolve by moving the surface. But when it moves the surface, it brings more precursor up to the surface again, which is then hydrolyzed. And so it keeps reestablishing, let's say, the imbalance of tension, and that's why it has continuous motion. There are lots of fluid dynamics responsible for this, like Marangoni-based instabilities. Not so important for this conversation, but what it might be important is that it has a low Reynolds number, which means viscosity dominates. What that means is when this thing starts to convect and the surface starts to move, there's friction between the droplet and the water phase, and that friction allows this droplet to swim basically through the water phase. It's the same way that we can swim through water by putting our arms out and making our palms flat and pushing against the water, forming friction. That's how we propel ourselves in water. So the surface of this is also forming some friction with water, and that's why this could move. So in this previous slide, I talked about the tension imbalance due to pH, so that basically is very sensitive to pH. In this slide, I talked about the actual the actuation of the droplet, which is due to this convective cell. And putting those together, we can then take a system where we can manipulate the motion by having a sort of free-running droplet here, where I add in a chemical gradient, which is a pH gradient. So it should be sensitive to this gradient. And so the droplet will move until it finds the pH gradient, and then it's able to basically change its motion so it could climb to the highest part of that gradient. So this is an example of chemical chemotaxis, droplet chemotaxis, artificial chemotaxis, because we can control the motion of these droplets because we understand enough about the mechanism of the motion. And biological systems do this all the time. Here on the right is a video of a dictyostelium cell, and someone here has a pipette which has a chemical stimulant in it or attractant called cyclic AMP, and this cell has receptors for this molecule, so it's able to receive a signal that where the cyclic AMP is in the environment, and it's able to change its cytoskeleton so it could actually move towards the source of that stuff. So this is what biological systems are good at. And our chemical systems on the left here can also do something similar. So here's a droplet, and we're putting in a chemical stimulant. And we can see that the droplet is very sensitive to that chemical stimulant, and it can change its behavior depending on where the gradient is. So this doesn't have proteins, it doesn't have proper receptors, doesn't have DNA or anything like this. It's just trying to minimize an energy landscape. It's trying to balance surface tension, as I explained. But still, in the end, what we've done is create a very simple chemical system that behaves like a chemotactic biological system. But again, simple principles here. So that's what we like about it. And because we can then show that this is like a sensor coupled with a motor, we then try to understand if these systems, because of these behaviors that just emerge from the system, actually are avoiding equilibrium, because these are chemical systems making their own waste product. And if they just accumulate waste products around themselves, they will stop. But they can continue moving if they actually can move away from their waste products. And that's what these things are doing. They're continuously making waste products and continuously moving away from the waste products, which then kind of makes them avoid equilibrium, but also maybe an example of a non-living system that is performing something that we might call self-preservation. This is the question here. So can we say these non-living systems are actually preserving themselves because of these properties? That's a question and a debatable subject. So that's my part. And Silvia. Yep. Silvia will take over. And of course, we welcome any questions, but maybe we can wait till the end. So please, Silvia.
Yeah. So I'm gonna now talk to you about the system with which I mainly worked with during these years working with Martin. And it's a super simple dynamic droplet system. And the reason why it works is was described before from Martin. So it's surface tension and variation and energy reduction. So in the specific, we will have in this system, may I?
Yeah, it should be working, yeah.
We have one body, that is one decanol, that is the body of our droplet. Then we have one environment, so water, that in this case contains some surfactants that are in this case decanoate. And we insert inside the system even one attractant. The attractant is a solution of sodium chloride. So these are really simple chemicals. It's a really simple chemical system, and we have the movement of our droplet as the one that Martin just showed you in purple environment, from a position A to a position B, in which we place the salt. So this system is really simple, but can be even artistically interesting, because it can behave even in different systems. We can use different colors to color the water phase, and as you can see from this video, we have the motion of the droplet of the body of decanol from its starting position to a second one in which you place the salt, and then we can play with it and make it move even using a variation of the pH. So this system is really simple, but it's nice because it can be, for example, used to transport objects. So as it's shown from this video, we charge the decanol droplet with a piece of copper wire. We moved it using the sodium chloride gradient. Then we destabilize the coupling between the droplet and the copper wire. We move the droplet a second time. We destabilize it. And as you can see from the video now, the last movement of the droplet is decoupled from the copper wire. So we can say that we have this system that is really simple, and it can be not simply used to move on its own, but even to transport objects. Now we wanted to use these characteristics and to try to transport even other things. So we talked about transporting living objects, so maybe living cells. And as we normally sell our packages, we want them to arrive sane and safe to its recipient. So we talked about use specific packaging to transport our living cells, and we decided to use arginate capsules. Arginate capsules are here in our system shown in white, yellowish. They're not so big, but they are present. And they are depicted by the arrows here. They are shown by the arrows. And we can see how, placing in the second point a salt gradient, our droplet transported the capsules move. And then if destabilized as before, using a specific solution with a high concentration of surfactants, they can be decoupled from the capsules, and the droplet can move a second time. So our system is not simply able to move inorganics, not living objects as copper wires, but even living objects as capsules containing cells. In this case, we transported different kind of bacteria. So we transported, for example, gram-negative and gram-positive bacteria, and both to, for example, E. coli and Bacillus subtilis. And if after the transports we pick up the capsules and we plate them overnight, we can show that the bacteria are still growing after the transport. At this point, so we showed in this paper that we can transport bacteria. But we wanted to transport not simply bacteria, but even eukaryotic cells. So we tried to transport mammalian cells. We were able to transport even mammalian cells, and this is the system that will transport them. So we always have another timed body, that is 1 decanol. We have a new timed attractant, that is sodium chloride. We have the cargo, that is in this case the alginate capsules with the mammalian cells inside it. And we have a different environment this time because mammalian cells are somehow sensitive to the decanoate and to the high pH. So we have one DMAM, that is this pinkish-like solution in which our capsules upon droplet are transported. And this is a video showing the transport of mammalian cells. So I said before, we have the droplet with the capsules inside this DMAM-like solution, and we were able to show that adding sodium chloride and varying the surface tension of our systems, we are able to move the droplet and the capsule with the droplet itself. So at this point, this is the final system that we created. So we have cells that are incubated, living cells encapsulated inside capsules, incubated for 2 days, because during these 2 days we've shown that capsule release some surfactants and that get entrapped in the border of the capsule itself, but even released in the water phase in which the capsules are incubated. This making feasible the coupling of the capsule with the droplet, and then in a second time the movement using sodium chloride gradient of the capsule and the droplet. Then the capsule can be picked up, the cells can be harvested, and viability is checked. Even mammalian cells survived. So what we found out that was interesting of this system is that mammalian cells showed one increased affinity, their capsule showed one increased affinity for decanol droplet. So we tried to study it better, performing some association tests between the droplet and the capsule. So here the droplet and here the capsules. And we were checking for the timing for which they were remaining coupled, until when the capsules was released to the bottom of our plate or becker, or where our system was placed. And we found that only a specific type of cells, so lung cancer cells, that were maybe producing while encapsulated some surfactants, had this increased association and increased liking for the association with the decanol droplet. So this is, as I said before, the test and the control by Trypan Blue and MTT test for the cell survival after the transport. All the names that you can see here are lung cancer cells, and all of them survived the transport. And in the specific, we then tried to better study the mechanism that was regulated this system. So the system that we used before to transport bacteria was already previously well studied, and it was the canoate, and with it we studied the variation in surface tension. And here is, for example, shown the variation of surface tension inside the canoate upon addition of sodium chloride. But we wanted at the same time to study why the chemotaxis was happening in this system. So if some surfactants were released too, and so if there was some variation in surface tension. And here is instead depicted the variation of surface tension upon sodium chloride addition to the water phase in which the capsules with Langer cells were incubated. So we tried to better analyze if this kind of variation was specific only of living cells, or if we're simply, or if the variation of surface tension and the compounds that were released surrounding the capsules and in the water phase was instead given from, for example, cell degradation or alginate degradation. And we find out that the change in surface tension that gives the association between the droplet and the capsule, and even the feasibility of the chemotaxis, is specific of living cancer cells. So the other thing that we found out is that we could use the water phase in which the lung cancer cells were incubated, even to transport lung cancer, other type of mammalian cells that are more prone to die after 2 days of incubation in DMM. So we created a new time alginate with inside it some this kind of water phase in which lung cancer cells were incubated. And we found that we can use it to do a second encapsulation of other type of lung cancer cells, such, for example, HEC293 and HS68. For who doesn't know, they are much more sensitive to pH changes, to lung incubation without DMM complete, and so without all the nutrients that they need. And so, for example, we try to transport them after 1-2 day incubation, but they die. And we found instead that using this double encapsulation of their capsule with the shell of the lung cancer cells, they can be transported, and they survive the transport. So to conclude, we have one system that is able to transport both 2 bacteria, yeast, and even mammalian cells. We have some cells that inside the capsules have one augment of their functions. In fact, the lung cancer cells did not seem to normally release this kind of surfactant-like compounds when simply incubated in DMM, but only when encapsulated before in alginate capsules. Then we found that some surfactant produced by cells can be used to increase the capsule droplet association and affinity, and even the chemotaxis efficiency. And that a shell containing surfactant that are released from these specific lung cancer cells can be used to transport cell lines that are normally not surviving the transport, and that do not survive detrimental condition of the chemotaxis. I think I've finished.
Yep, that's the end.
Great, thank you both for your presentation. It's now time to move into some Q&A. And just for those of you who are not here participating in the live session, if you're watching this tomorrow, because it's the middle of the night for you, it's still possible to ask questions on the seminars.alife.org. If you find the page for this talk, there's a forum at the bottom where Martin and Silvia will check in at least regularly for the next couple of days, just to see if there's any questions that need to be that have been asked there and that they might be able to answer. So if it's all right, I'd like to open the floor now for some questions. We've got several people that have arrived during the talk, and maybe I'll kick off with one question if that's all right. And so my question has to do with other environments for trying to move to use these kinds of droplets to move things around. So obviously the first thing that comes to my mind when you're talking about using oil droplets to move living cells around is some kind of medical application down the road, right? The idea of using these droplets or something like them to go deliver drugs to some nasty tissues or to some other, or to remove tissue from wounds or things like that. But of course, the environments that you're looking at are very different from the organic ones. They're sort of relatively big open spaces, is what I'm thinking, right? You've got this big petri dish, and you've got this relatively large scale droplet. And I was wondering if you've even just played around with or explored any other kinds of environments, like, I don't know, I was thinking capillary tubes, or even surfaces, but that are organic, like, I don't know, some skin or something with some droplets that would be moving around on top of that. Have you gotten to that stage, or is that still something for the future?
Okay. Yep. Good question. We have we've done a little bit of so I'll answer your second part first. We've done a little bit of work with different environments, not skin, but we've done these droplets work in a smaller scale. We were able to do different kinds of media that are. So one of the most interesting one was when we were working with. Instead of a glass dish, we work with a glass fiber network. Okay. So basically a kind of mesh of glass fibers, and we can see how the droplets are moving and behaving within this network. So that works. We're also able to control some of the motion of these. They basically branch out like an amoeba into these networks. It's beautiful video, actually. I can show you sometime.
Just to clarify quickly, Martin, is that them inside the tubes, or is that like a forest of shards of glass, so to speak?
Like moving through a jungle. So
okay.
they're moving through the open spaces of the jungle. And they basically break up. So then you have populations of droplets that are moving with fluid dynamical motion through these things. So that works. But most of this is not yet explored, Matthew. Yeah. And I now want to just say a word about the first part of your question, because I think this is the point is, yeah, there can be things like medical applications, but I think the bigger picture is, and this was discussed quite a lot in ALIFE in Tokyo when we were there, is, what's going to be next? How can artificial life impact the world, such as artificial intelligence seems to have done? And I think one of the ways to explore that is trying to say, okay, well, we have all these great ALIFE tricks, basically, different models. We have in silico stuff. We have chemical droplets and other things. What do we do with this stuff? How is that going to impact the real world? And so that's what Silvia and I have been working on. This is just an example of, well, how do you bring the artificial life world and the living world together? And when you crash them together, some things don't work. I mean, the chemical systems just start to kill the cells, for example. Okay, so let's solve that problem. And then we found, oh, well, not only does it solve that problem, but the living cells, if we keep them alive, will supply the chemistry we need to run the droplet experiment. Oh, that's cool. So, I mean, how do you bring together these two worlds, the biological world and the artificial life world, is the general principle that we're dealing with here. Whether this has medical applications, it'd be cool for people, collaborators to try. But I think we're interested more in these bigger issues.
Cool. I really appreciate that response. I mean, I think it's always an interesting conversation to think about the different ways that artificial lifestyle research can be brought to bear. So it's just as you're saying, investigating that interface between biology and complex systems and so on. And another area where I think ALIFE style research can make lots of interesting contributions is in sort of the philosophy of biology and the philosophy of cognition. And along those lines, there's another question I had for you, which was brought up by your talk, which has to do with, at one point, Martin, you said that your droplets don't have a proper sensor the way a biological individual might, the amoeba or amoeba-like organism that you're looking at, that you have showed the animation of, video of. But I wonder why you say that, and I'd like to push back, and I'd like to say that your droplet does have a sensor. Why do you demote the status of its sensor?
When I said that, I had a little bit of feedback in my brain said, "Oh, that was a stupid thing to say." And how did you pick up on that? That's pretty good, Matthew. Yeah, that's right. I think that was wrong for me to say, but I was trying to make a point. Maybe I can clarify is that biological systems have specific sensors for different environments that are fairly sophisticated, right? So these are complex molecular structures that are evolved in 3 dimensions and also kinetically to recognize different types of ligands and things. And this is not that. I mean, this is just, these are just simple molecules that self-assemble and have 2 states. And these 2 states are controlled by the pH, and then therefore the pH controls the system. So they are sensitive to pH. They're pH sensors. They're proper pH sensors. They're just not necessarily the way biology does it, right?
Yeah, they're not as complicated. Yeah,
they're not as complicated.
I can get on board with that message for sure. Do we have any other questions from other people that are here in the live session? That anybody want to ask anything? I had a couple of questions here. Oh yeah, and it in fact relates to your last comment, Martin. So, and this is, so my question is, from your perspective, having worked with these systems, Martin, I think it was the first slide you showed, there were these different aspects of life that you mentioned: reproduction, motility, agency. You know, it's this sort of tree of different phenomena that you're interested in studying. You've now gained a lot of experience by exploring those phenomena in a variety of chemical systems. And I'm curious what you've learned about those systems in terms of which seems the most likely to have preceded the others. Do you have any comment on that? A lot of people on the origin of life as being about the origin of evolution. And so a lot of attention has been focused on how do we make molecules that are capable of reproduction or copying themselves. But maybe that's actually something that came after a lot of the other phenomena that you're looking at. Do you have strong opinions on which of those came before the others?
So, yeah, I do. I think that several of these things are easily accessible. And I was surprised that self-motion is among those. I thought, the ability for living systems, the way that living systems move seem very complicated. And the fact that you can get a non-living system to move so well, that's quite interesting. So I think that was a surprising one. What is not so surprising is maybe more of the reproduction stuff, because every day in your kitchen you have different kinds of liquids that are evolving in the sense that they are fusing together and splitting apart. Any kind of emulsions you have. Emulsions include mayonnaise, ice cream. When your freezer breaks and your ice cream melts and then you refreeze it, it turns into a different state. And that's what we're talking about here. So these kinds of transformations of fusion and fission that could lead to replication cycles seem rather intuitively easy to do. Okay, then it becomes a challenging part. So agency is really challenging. Even information embodiment, I think, is challenging in these systems. So how I believe these things have information. What is that information and how is it there? And how do we, if we can, quantitate it? That's really hard. So what does that mean? Does that mean we have Omer, who's probably listening to us, has done a lot of work with compositional information. Does the composition of chemicals at a certain point of time give us some information that is embodied in these systems? And if so, how important is it? Does it matter? Does the system, in a sense, utilize that information? Or is it just kind of irrelevant? Maybe just a historical record that doesn't do anything? So the questions then become, how do you make experiments to figure this out? For example, can you do an experiment that shows that the history of a droplet, what it went through before, will then affect the behavior in the future? This would be a cool experiment, So that's what I think where the simple stuff is. So the point is, I think when we learn how to do the right experiment, perhaps all this information is already embodied. We just don't we don't do the right experiment, or maybe we don't even think properly about it. Maybe from a Droplets point of view, if it had a point of view, it's already all there. We're just not smart enough to figure it out yet. And then the other thing is, you just mentioned briefly the origin of life, and I didn't talk about that today, is I think it's also kind of an interesting perspective that these experiments can give us on the origin of life, because when we use, like Silvia and I use these pure, simple chemicals from a chemical supply company, that works great. If you go beyond that in a lot of chemical experiments or origin of life experiments, things fall apart. They cannot deal with tremendous complexity, which I think is more of a real world situation. These droplets can deal with tremendous amount of complexity. So we've done some studies where we've taken a population of molecules through this combinatorial explosion, and we use those to fuel the motion and chemotaxis of these droplets. So the droplets can deal with huge amounts of chemical complexity. Maybe that's more relevant to origin of life studies. Now, whether a droplet has anything to do with evolving a cell eventually, who knows? They think they're pretty far away. I mean, the very nature of a non-aqueous droplet is very different from what a cell is, which has an aqueous core and a boundary of lipids and things. But they're in the same family, this extended family. So we just have to think about what these results mean. They're easily accessible solutions, but what are they? How meaningful are they?
Cool. Yeah, super fascinating. Thank you. I'll open up again the floor for any further questions.
I think we have a hand up.
Oh yeah. I don't see that. So call on them, Martin, if you can.
Okay. So we have fellow Gister is registered here. I see has a hand up.
If anybody wants to ask a question, go for it. You might have to unmute yourself.
Yeah, there we go.
I've got one. I'm not sure if that's my hand or not.
Yeah, it's your hand. Yes.
Okay. I couldn't tell that it was successfully raised. So I've got a very basic question. I'm actually not entirely sure I grasp the significance of the second part of the talk and the moving around of cells, but just in terms of the very base, the first half of the talk, the very basic motion, if I could sort of echo back what I think I understood about how it works, you could tell me if I got this right. And I'm probably reading more stuff into it. I'm going to say stuff you didn't say because I can. Like you didn't use the word membrane, but I'm going to go ahead and do that because it looks like a membrane to me.
Yeah.
It looked, when you called it with the convective cell, I think, it looked, what I think I imagine happening there is you have different tension on one end of the cell than the other. And so there's kind of like a force to maybe add membrane to one side and remove it from the other side. And that stream we saw going down the middle of the cell was actually the movement of membrane to redeploy it to the opposite end.
Yes.
And then, okay, and then you also said that there was friction with the water. So what I'm imagining is that by adding that membrane to the front end, it's actually kind of tractoring through, like crawling through the water as if it were more of a rigid substrate from its own point of view.
Mm-hmm. Yep, I think your understanding is correct. It's
okay.
The scale, the physical scale of these experiments will predict that there is viscosity that dominates. So basically it's feeling that the droplet is able to feel that the water is viscous, so it's able to then create friction with it. Yes.
Yeah. Okay. Interesting. Thank you.
So when you say that, Martin, when you say, because I was wondering about that earlier, you said the Reynolds number is low and viscosity dominates, but I wasn't sure what you were saying it dominates over. What would it mean if the Reynolds number were high? Let's say you had exactly the same system, but the Reynolds number were high. Viscosity is not dominating, so what is?
So then you would have, well, you would have actually a few different kinds of effects, actually. One is you probably would not be able to sustain a convective cell because it's just the wrong length scale. And so, and then what it would be is that you would have no, you have nothing to push against. If basically the viscosity goes very, very low on the system, You can move all you want. It's like, okay, it's right now, Matthew, you can try to swim through the air.
Right.
Good luck.
Right. Yeah.
Yeah. So
Okay.
that's the problem. That's the problem with the system.
So you might still see flow inside the convective cell. You might still see the convection, no? But it's just that it wouldn't propel the cell through the water at all, or no?
Also depends on which variables we're changing. You could still get a convective cell that is not moving. Yes, that's right.
Okay.
Mm-hmm. Yeah.
Cool. Great. Okay. Well, if there's no additional questions, I think we might wrap things up there. And as I said before, there's an opportunity to ask more questions online. Thank you both for your talk. It was really nice to hear you speak about your research and to hear about it again. Every time I've heard you talk about this a couple times, but every time I hear a talk, I learn a bit more. And so I very much appreciate it. And I'm sure the ALIFE community will as well. I'll see you guys virtually in Montreal, virtual Montreal for ALIFE this year. And thank you again. It's really been a pleasure to have you. So thank you. And I will end the stream now.
Thank you. And thank you for coming. Thank you for participating today.